E-Biology class presented by Fatima Convent High School. Myself, Dipali. Chapter 6 Tissues. In this session, we are going to study about the plant tissue. We already understood what is a tissue and how the plants and animal tissue are different. Now, what is plant tissue? Now, we all know that tissue means a group of cells performing specific functions and have specific structures. So, plant tissues are tissues which are performing specific functions and have specific structures are called as plant tissues. And according to the dividing capacity, these plant tissues are classified into two main groups: meristematic tissues and permanent tissues. Now, what are meristematic tissues? Now, according to the dividing capacity, if the plant tissue keeps on dividing throughout the life, it is called as meristematic tissues. Let us see this with the help of an activity. We are going to check the growth of an onion bulb. Now take two glass jar and fill them with water. Now take two onion bulbs and place one on each jar as shown in figure. Observe the growth of roots in both the bulbs for a few days. Measure the length of the roots on day 1, day 2 and day 3. On day 4, cut the root tip of the onion bulb in jar 2 by about 1 centimeter. After this, observe the growth of roots in both the jars, measure their length each day for 5 more days and record the observation in tables, like the table below. Day 1, Day 2, Day 3, Day 4, Day 5. Jar 1, Jar 2. Now, the growth of plant occurs only in certain specific regions. This is because the dividing tissue, also known as meristematic tissue, is located only at these points. Depending on the region where they are present, meristematic tissues are classified as apical, lateral, and intercellary. So, what did you observe? We observed that which of the two onion had longer roots and why? Do the roots continue growing even after we have removed their tip? Why would the tip stop growing in jar 2 after they are cut? Now, the answer to this question is the bulb 1 will have longer roots because at the tip of the roots, meristematic tissue is present and it keeps on growing continuously, and because of that, the length of root keeps on growing. Whereas in group 2, that is in jar 2, the roots will stop growing because we removed the tip of the root, and because of that, the meristematic tissues are removed, and so the root will not grow any longer. Why would the tip stop growing? In jar 2, as we said, the meristematic tissue is no longer present in the roots and so it is removed. Now the growth of plant occurs only in certain specific regions, and this is because the dividing tissue, and this dividing tissue is meristematic tissue and is located only at these points. Depending upon the region, these meristematic tissues are again classified into three groups. They are apical meristem, lateral meristem, and intercellary. New cells produced by meristematic tissues are initially like those of meristem itself, But as they grow and mature, their characteristics slowly changes and they become differentiated as components of tissues. Now first let us get familiar with few parts of any plant. Now if you take any stem, then you must have observed these parts. The tip of the stem is called as the apex. The place between the two leaves growing is called as internode. These all places are called internodes. And the point from where new plant leaves or new flowers grow are called as nodes. Are called as nodes. So if the tissue is present at the apex, it is called as apical meristem. If it is present at the node, it is called intercellary. And if it is present in the internode, it is called as lateral meristem. And all these three have different functions. What are those functions? Now being at the tip, it will help in the growth of length of the roots and stems. Intercellary meristem helps in the growth of new leaves and flowers. And lateral meristem allows the girth of the plant to increase by adding more and more layers of meristematic tissues. So the apical meristem is present at the growing tip of the stems and roots and increases the length of the stem and the roots. The girth of stem or root increases due to lateral meristem cambium, which is the other name used for lateral meristem. So, the intercellary meristem seen in some plant is located near the nodes. Now, how are the cells of meristematic tissues? So, the cells of meristematic tissues are active and they have dense cytoplasm because they have to constantly grow. Thin cellulose walls and prominent nuclei. They lack vacuoles because they don't perform the function of storage as they constantly multiply and grow. So, you can say the characteristics of the meristem is or meristematic tissues is cells are active, have dense cytoplasm, thin cellulose walls and prominent nuclei and absence or lack of vacuoles, right? That is the characteristic of meristematic tissues. Now let us see what is permanent tissues. Now we know that these meristematic tissues constantly keep on multiplying or dividing. But what if it stops dividing and multiplying after some time? Then they just take up a specific role and lose the ability to divide. As a result, they form permanent tissues. So, the permanent tissues are nothing but meristematic tissues which have lost their ability to divide anymore. This process of taking up a permanent shape, size and function is called differentiation. Differentiation leads to the development of various types of permanent tissues. Now, according to their complexity, they are divided into two: simple meristematic tissue, simple permanent tissues, and complex permanent tissues. Now, let us perform an activity again to see what are these groups of permanent tissue. Take a plant stem and with the help of your teacher cut into very thin slice or sections. Now stain the slice with safranin. Place one neatly cut section on a slide and put a drop of glycerine. Cover with a cover slip and observe under a microscope. Observe the various types of cells and their arrangement. Compare it with figure 6.3. Now answer the questions given below. Then you will observe, are all cells similar in structure? No, it is not possible as you can easily see in the diagram. They have different structures. How many types of cells can be seen? Then we can see epidermis, collenchyma, parenchyma, phloem, xylem, which is also called together as vascular bundle. Can you think of reason why there would be so many types of cells? Because each cell is performing different specified function given to them. And because of that, they need to have special structure and get differentiated into different forms so that they can carry out their function easily. We can also try to cut sections of plant roots. We can even try cutting sections of roots and stems of different plants. So, simple permanent tissue, that is the first type of permanent tissue. A few layers of cell beneath the epidermis are generally simple permanent tissues. They are called as parenchyma. In most common simple permanent tissue, it consists of relatively unspecialized cells with thin cell wall, that is the characteristic of the parenchymatic cells. They are living, they are usually loosely arranged, thus, large space between the cells, intercellular spaces are found in these tissues. These tissues generally store food. In some situations, it contains chlorophyll and performs photosynthesis, and then it is called chlorenchyma. In aquatic plants, large air cavities are present in parenchyma to help them to float. Such parenchymas are called arenchymas. The flexibility in plant is due to another permanent tissue, collenchyma. Chyma means tissue and the prefix represents what its function is. It allows bending of various parts of the plants like tendrils and stems of climbers without breaking. It also provides mechanical support. We can find these tissues in leaf stack below the epidermis. The cells of these tissues are living, elongated and irregularly thickened at the corners. There is a very little intercellular spaces. So you can see the three different types of parenchymatic cells. The first one is parenchyma, second is collenchyma, and third is sclerenchyma. Yet another type of permanent tissue is sclerenchyma. It is the tissue which makes the plant hard and stiff. We have seen the husk of a coconut. It is made up of sclerenchymatous tissues. The cells of these tissues are dead. They are long and narrow as the walls are thickened due to lignin. Often these cell walls are so thick that there is no internal space inside the cell. This tissue is present in stems around the vascular bundle, in the veins of leaves, and in the hard covering of seeds and nuts. It provides strength to the plant. So, did you observe how the tissues are classified into various sections? According to what we studied till now, let us just revise what we did. So, we know that plant tissue. Now, plant tissue is of two main types, meristematic and permanent. Meristematic are of three types, apical, intercellary and lateral. We are also very much clear with the function. Meristematic tissues are constantly dividing tissues. Apical meristem is present at the tip of the stem and roots. Lateral meristem is present at the nodes and intercellary is present at the nodes and lateral meristem is present at the internodes. We also know the characteristics of cells of meristematic tissues. Now, permanent tissue is divided into two broad groups, simple and complex. Now this simple is divided further into parenchyma. If it is performing the function of photosynthesis and has chlorophyll in it, it is called Claren- chlorenchyma, arenchyma, where it helps in increasing the air volume, and last you have the sclerenchyma, which gives rigidity and support. Are you understanding? These are the basic classifications of the tissues, that is simple permanent tissues. The parenchyma, chlorenchyma, arenchyma, collenchyma and sclerenchyma. Now let us see what are the guard cells. Take a freshly plucked leaf of reo, stretch and break it by applying pressure. While breaking it, keep it stretched gently so that some peel or skin projects out from the cut. Remove this peel and put it in a petri dish filled with water. Add a few drops of saffron in. Wait for a couple of minutes and then transfer it onto a slide. Gently place a cover slip over it. Observe under microscope. What you observe is the outermost layer of cells called epidermis. The epidermis is usually made of single layer of cells. In some plants living in very dry habitat, the epidermis may be thicker since protection against water loss is critical. The entire surface of plant has an outer covering epidermis. It protects all the parts of the plants. Epidermal cells on the aerial part of the cell often secretes a waxy water resistant layer on their outer surface. This aids in protection against loss of water, mechanical injury and invasion of parasite fungi. Since it has a protective role to play, cells of epidermis tissue form a continuous layer without intercellular spaces. Most epidermal cells are relatively flat. Often their outer and inside wall are thicker than the inner wall. So the next layer is epidermal tissues or epidermal cells, which is the outermost layer of the plant body and which has the characteristics as shown. So that is called as epidermis. Now in the epidermis there are small pores, and these pores in the epidermis are called stomata. Stomata are enclosed by two kidney shaped guard cells, and they are necessary for exchanging gases. With the atmospheric transpiration, loss of water in the form of water vapor also takes place through stomata. Recall which gas is required for photosynthesis. Find out the role of transpiration. So for photosynthesis we need CO2, but for transpiration we need O2. So the epidermal cells of the roots, whose function is water absorption, commonly bears long hair like parts that greatly increases the total absorptive surface area. In some plants like desert plants, epidermis have a thick waxy coating of cutin, chemical substance with waterproof quality on its outer surface. Can we think of a for this? In the outer layer of the branch of a tree, different forms of outer layer of young stems. As the plant grows older, the outer protective tissue undergoes certain changes and a strip of secondary tissue located in the cortex forms the layer of cells which constitutes the cork. So that is the outermost layer, the thick bark-like structure of the plant. Cells of cork are dead and compactly arranged without intercellular spaces. They also have a substance called suberin in their walls that makes them impervious to gases and water. Did you understand what are simple permanent tissues? In next session, we are going to study about complex permanent tissues. Thank you.